animals. The third one is how we can use deep learning to save lives. So we worked with a pharma client who was who's looking for help with drug discovery. So there are generic players who have to come up with drugs which mirror the innovator drugs. There are patented drugs which are in the market, and the generic companies come up with similar drugs, and they do a comparison. How close is this generic drug to the innovator drug? And a key process in that is the drug characterization, where they have to study what is the composition of the cells when you administer it to a patient, and you take out the cell count and find out what is the distribution. Is it very similar for the generics and for the innovator drugs? The way this is solved today is it's largely manual. People take these microscopic images which look like this. And there are multiple shapes which you see here. There are circular ones, square, and approximate other shapes. There are 4 shapes which scientists look out for here. And each of these shapes have to be counted. And to ensure a similarity of this drug with the innovator, they have to do the composition verification. Assuming the shape 1 is 60%, you check whether it is a similar composition for the innovator drug also. So this, they have to do it across hundreds or thousands of images. And it's a largely manual process, painful. So when we started working with the client, we wanted to see what approach would work here. At the outset, though, it looks very different, but it has similar characteristics of a crowd counting problem. So here again, you have those density clusters, certain portions crowding out the cells. And then there is a challenge of occlusion. There are some cells which overlap and come in front of the other one. And so on. There are these images which have low light, dark light. You'll have to do some contrast improvement. Those challenges exist. So when we saw the similarity, we took up a similar cascaded CNN and tried to apply there. But remember, this was something which we were directly doing it for an enterprise client with some data that they had, and they were trying to deploy this to production. So unlike the earlier cases, we ran into some stumbling blocks. What were they? Data volume. So far, we are talking about 10,000 or 100,000 images labeled, and thanks to the volunteers or crowdsourced efforts. Whereas here, we are talking about images which either don't have labels, or in some cases, you have just hundreds of images. So it could be 400 or 500 images which have been manually labeled by the people. Usually people, when they count it, they may not label it painstakingly. So that's the labeling that they would naturally do. Whereas for the model, we'll have to zoom in, place the dots at the exact position, identified by the different shape, and do this many times for different images. So this kind of labeling did not exist. And there are fuzzy entities. Earlier, we talked about the faces or objects, or in terms of even people having relatively standard shapes and sizes. Whereas here, we are talking about a circle which could approximately look like a circle, or could be like an oval. All of these are comfortably combined together into one single bucket. So when it is tough for humans, how would a model which is trained on strict circular shapes be able to learn the other thing? So that is the challenge which we were running into, where the entities are fuzzy. How do you ensure that the model learns the approximate boundary? And then another issue was the boundary of the entire shape. You can approximately make out a circular boundary for this shape. So this is the plate on which the sample is placed and put under the microscope. So anything outside of that circular boundary is not useful. In fact, we should avoid counting it. So it could set the model off in a completely opposite direction. So how do you identify that boundary? By the way, it's not a perfect circle. The way it comes out in the microscope, it could be in any of various sizes and shapes. And it is always not at the center. It could be offset. So we had to try out a variety of approaches. Eventually, we used this approach of edge detection. So we used the RANSAC model, random sample model, which ran through multiple iterations, tried to detect the edges of this shape. And this is basically an outlier detection approach, where you find out what are the boundaries, and whatever lies within these boundaries are data points which we're interested in. Anything outside of that is an outlier, a standard outlier approach. So when we ran this through multiple iterations, it started detecting the edge, and we approximated it to be a circular shape. And with that, we were able to find that this model fairly works well. The RANSAC model fairly works well. and we were able to clip out and apply a circular mask. So the model returns the XY coordinate, what is the center, and it returns the radius. So with that, you're able to draw a circle around it, clip out that portion, and that becomes a focus area. And other challenges, earlier we talked about counts and we said we had 500 people, instead the image predicts 550. So about 10% or 5% in other cases, which is okay. We can go with that approximation. That doesn't work in many business scenarios. So here the challenge was, yes, you've got it to a close enough number, which is good, but we can't go with that. We need to correct it further. So the question was, how can you enable us to work on top of it, which is a classic case of human augmentation along with AI. And you let the human edit and alter the results, which can be submitted finally. So we had to figure out how to do this. I'll come in terms of how we solved it. And the other critical requirement was integrating this with the business workflow. There's a certain way that the researchers were used to working. We had to design an application, a UI, and a workflow which worked along with that. It cannot be a standalone thing, in which case the adoption would not happen. So that was something which we had to plan for, on how you can humanize this intelligent and make it useful for people. Finally, we had to live with and see how we can work with low accuracies. So given all of these challenges, you can only expect that the errors are likely to be higher and the accuracies are lower. So how do you work with that? And importantly, how do you communicate and convince clients to go with that? So here's the approach we had taken. Firstly, we cleaned the data. So we identified what are those images which have a completely off contrast, and then we clean them. And we label the images. So like I mentioned earlier, we assume that whatever has already been used by the scientists can directly be used as labeled images. It just didn't work. So we had to go back and spend a lot of time on manually annotating the images. Even for hundreds of images, you can imagine it's a huge effort. And then the boundary identification, like I mentioned, ransack model. After a few iterations, it worked well. And then we put this through all of the images which come after the stage are put through the counting model. So now you can see that the counting model starts picking the cleaned image, splits it into the same 3x3 patches. You have 9 patches, and each patch goes through these 2 stages of high-level prior to do this approximation, classification into one of 5 or 10 buckets, and the second stage, which does the density estimation, the Gaussian density estimation. So similar approach as we had used earlier. And with this pre-processing, it started giving us results. And at this stage, we were fairly excited, and we said, yes, we have solved this problem. And that's when the challenge came in that, like I mentioned, even if this 50 or 60 images are not right, we are not happy with that. We have to change that for us to start using the solution. So we had to go back to the drawing board, because we had just the density map. How do you get the coordinates of each of the shape from a density map? There are no standard approaches for that. So after some back and forth, we settled on a contour detection. So given a heat map, there are certain portions, you've seen that there are these colored portions and other edges which it detects. So from those contours, can you identify the coordinates? You pass through the entire image, and then like line by line, row by row. And then wherever you notice that there is a speck which the model has placed as part of the density estimation, you pick that up, identify the position XY coordinate. That is most likely the position of one of these shapes. So you'll have to do that for the complete image and then pull it out. And then we had to match this count, because this might be giving us a count of 100, whereas the model might be giving us a count of 500. And only 100 of these shapes will be labeled. So the model falls flat there. So we had to do an iterative approach where we ran through this multiple times, and we started converging these numbers. Whenever both these numbers, 100 and 500, they start converging, that is when we can come to a conclusion that the numbers are pretty close. So that was our approach, contour detection and iteration to find out the XY coordinate, and then start labeling them back onto this image. So once we did that, we had to show this on a UI to the users. So we built a UI. This is what it looks like. We let them upload an image or a bulk set of images, so you can just point it to that location from where the microscopic images are stored. And after you save it, the cleaning and model counting gets initiated. So the model automatically runs on all these images which have been loaded, and it creates an annotation like this. This is the model-generated annotation. It detects the shapes. You can see the 4 shapes, type 1, 2, 3, 4, circle, square, cylindrical, and all others. And you can see the dots are the blue dots, where you can find out that these are roughly the circular shapes, and it's counting about 383 of these. And based on the contour detection, it has placed this dot. We aim for the center, so it at least stays within the shape. And this is presented to the researchers. So now, once they get into this interface, they can scan and validate this. Assuming they find that one of those images is misclassified, it has to be a square, it is not a circle. They click on that dot, and you see this pop-up come up. They can just reclassify, saying it's not type 1, it has to be type 2. So then and there, you can make this reclassification. Alternately, if you find that there has to be a new shape which has to be added, you see the controls on the top right. You have a plus button which lets them annotate then and there. Or if you want to clear off an image because it's just not a cell, you use that eraser to just clear off this image. After you do all of this, which might take about 5-10 minutes, then you can save and submit it. So this completes the business workflow. The model has taken it to 80 to 90%, and then with a human validation, we are able to complete the loop. A classic case of AI plus humans working together, which is what we had envisioned. And with this, the business can start using these results. And additionally, this also has the added benefit of improving the annotations for us. So the next time we have images which have been additionally curated by the users. So we can take these images and do a retraining of the model the next time. And the model results will start getting better. So over weeks and months, the accuracy would definitely improve.